Thank you everyone for joining us today. My name is Sabrina Severin and I am the Ocean Health Education and Conservation Coordinator here at Diventures. Again, thank you for joining our Coral Restoration in Curacao webinar with Dr. Dirk Petersen from SECORE. Dr. Petersen is the founder and executive director of SECORE International, a nonprofit organization that works to sustainably restore coral reefs worldwide. Since 2002, Dirk has developed SECORE into an internationally committed and leading institution in the field of research, technology, and development in the implementation of new restoration methods for coral reefs. After studying biology at the Ludwig Maximilian University of Munich, he did his PhD at the University of Duisburg-Essen and carried out fundamental coral breeding research in the Marine Biological Laboratory of the Rotterdam Zoo in the Netherlands and on Curacao. Dirk is an alumnus of the General Federal Environmental Foundation and lives with his family in Bremen, Germany. Dirk, take it away.
Thanks, Sabrina. So let me get the screen shared and then the PowerPoint going. All right, perfect. So I would like to, first of all, thank Diventures for hosting this webinar to allow me to share some of our work with you and how we help conserving and restoring coral reefs. I'm also excited to share some insights with you about the upcoming dive expedition to Curacao. SECORE is a nonprofit organization for coral reef conservation and restoration. SECORE stands for Sexual Coral Reproduction. We are headquartered in Ohio and have offices in Miami, as well as in Germany, Curacao, and Mexico. Our mission is to create and share the tools and technologies to sustainably restore coral reefs worldwide with the vision of a world where breathing coral reefs support healthy oceans for future generations. I guess most of you, if not all of you, are avid scuba divers. So I don't have to tell you that coral reefs are in a crisis. I guess most of you have already seen bleached or dying coral reefs. 1 billion people depend on coral reefs, serving as an important food source, protecting shorelines from storms and erosion, and serving as an important economic income through tourism. The International Coral Reef Society, which represents science internationally, has recently stated in a policy paper that we have 10 years left to take action to saving coral reefs. It is very important that we continue with protecting coral reefs through marine protected areas or sustainable fisheries, and most importantly, that we immediately address the climate crisis. But we know by now that all of this will not help to save coral reefs without active interventions. Most importantly, coral reef restoration. And I just want to point out that M. Miller, one of the authors of the paper, is in bold because it's Dr. Margaret Miller, who's our research director. However, current restoration efforts are too labor and cost intensive, since until recently, the most common practice was to manually produce and outplant coral fragments, which involves gluing every single coral on the reef. As a consequence, restoration efforts are limited to less than a hectare, while the degradation occurs on a scale of hundreds to thousands of square kilometers. Therefore, the most urgent question is how we can increase cost efficiency and to decrease labor to get restoration to scale. SECORE is in a unique position to address the challenge of scaling coral reef restoration. We carry out rigorous scientific research to understand how corals reproduce and how we can breed and rear them to produce large numbers of coral juveniles for reef recovery. Through engineering, we transfer this knowledge into applicable technologies and provide the necessary training and technical hardware to reef restoration practitioners through an international capacity building program. Corals reproduce by releasing their eggs and sperm during a few nights every year around full moon. The egg and sperm cells drift to the water surface where fertilization occurs. After a few days, free-swimming larvae develop that descend to the reef to find a suitable place to settle and turn into an initial coral polyp. After a few years, the coral baby turns into a young coral. Every step in the early life history must be passed for successful reproduction. Unfortunately, more and more species and populations do not manage to successfully reproduce any longer due to local stressors and climate change. Without reproduction, coral populations cannot recover. In addition, sexual reproduction is key for new coral populations to adapt to rising water temperatures and increasing their resilience. We at SECORE, together with our partners, help corals to reproduce again and to better adapt to climate change. We collect eggs and sperm during natural spawning events with using special plankton nets. We then carry out in vitro fertilization and rear the larvae in special enclosures. The larvae settle on special substrates, which we then seed on the reef without further attachment. The goal is to have one surviving coral per substrate. This young coral has by then overgrown its substrate and has firmly attached to the reef. It will then start joining the coral's annual spawning, thus closing the life cycle. The Alcon coral on the right is about 5 years old. We collect gametes from the corals by putting nets over the colonies a few minutes prior to they will spawn. Released eggs and sperm cells are collected in small samples at the top of each net, small sample tubes, of course. We routinely in vitro fertilize the eggs by simply mixing the different samples together. In this way, we usually obtain fertilization rates above 90%, often close to 100%, compared to less than 1% in the wild. The picture shows a participant of a training event in Mexico. The developing embryos are then transferred in large floating devices that we call cribs. This is a relatively simple way to rear thousands of coral larvae and babies in remote areas without the need for cost-intensive laboratory facilities. This crib on the picture was used for restoration work in Bonaire, one of the 10 countries we are currently active in the Caribbean. We developed special substrates on which the coral larvae settle inside the cribs. The substrates are designed in a way to increase survival of the young settlers and to self-stabilize on the reef without the need of manually attaching them. Here you can see a coral substrate a few months after it was outplanted on the reef. The little brown dots are baby corals. Our aim is to settle more than one larvae on a substrate, since the initial mortality rate is very high. With this, we aim to ensure that at least one coral will survive the most fragile initial life stages. Last but not least, the coral babies are placed on the reef by divers, wedging the substrates in crevices, like here done by Kelly in Curacao. We train coastal communities throughout the Caribbean to use the knowledge and technologies that we develop together with our research and engineering partners. We're currently active in 10 countries throughout the Caribbean. We have an office in Miami and research centers in Mexico and Curacao, at which we carry out scientific research, develop some of the technology, and carry out some of the partner training. We established the first center in Curacao more than 10 years ago. And I, as you already know, have a very special connection to Curacao. Since I carried out all the field work for my PhD thesis in Curacao about 20 years ago, I was studying coral reproduction to understand how corals can be bred for conservation and restoration. This was the time when I got the idea to start SECORE. Spending many hours underwater in the freezing reefs of Curacao was the highlight of my science career. Coral spawning is still like a miracle to me, and I enjoy every opportunity to get in the water to observe it. I'm thrilled about the opportunity to show those of you who are interested in joining me for a trip to Curacao this October, the beautiful reefs of Curacao that were my second home. The work that we are doing to date to rehabilitate coral reefs and I will take you out to my favorite reef to witness coral spawning during a night dive. We are excited to be collaborating with Diventures as part of this trip, because Diventures has been a long-term partner of SECORE and a strong advocate for coral reef conservation. Guests who join us on this Diventures trip will stay at Lions Dive, which is closely located to the Curacao Sea Aquarium, where I was based during my PhD thesis in Curacao. I spent hundreds of hours underwater at the Fringing Reef, right in front of the Sea Aquarium, one of my favorite dive spots in the Caribbean. We will dive during the day to enjoy the biodiversity hotspot and to examine the coral babies that my team has outplanted since a decade to further our research. And last but not least, we will enter the water at night to hopefully observe some coral spawning. We will also visit our research center at the local Camarby laboratory and field station of Curacao. We have some research going on at the reef right in front of Camarby, as well as in Camarby's laboratory facilities. You will meet Valerie Chamberlain, who did her PhD with SECORE and Camarby, and is now a SECORE staff scientist based at the Marine Field Station. As well as Kelly Lattenauers, who oversees some of the restoration work on Curacao and in the region. Last but not least, Tanya will join me and accompany us for the whole week. Tanya is coordinating our training and implementation program. She works closely together with our partners and regularly visits the partner locations across the Caribbean to provide advice on coral spawning and restoration work. Tanya is based in Mexico. Let's talk about coral spawning a bit, how it works. For most species, the reproductive season is very, very short. It spawns only a few nights a year and actually lasts only a few hours a year. So how do coral know when to spawn? Let's imagine you're a coral and you're stuck on the bottom. It's not like when the time comes that you can walk around to go find a partner and do some sort of parade to convince him or her to spawn together. On top of that, the coral sperm and eggs are only viable for a few hours. So if you spawn a little bit too early or a little bit too late, then you literally have missed the party and wasted a whole year of investment in reproduction. There are 365 days in a year for a total of almost 9,000 possible hours. So how do you pick the right time? Most corals spawn during the warmest months of the year and will schedule the date of the month based on the lunar cycle. The exact time of spawning will be geared by the sunset time. So for Curacao, Valerie and her colleagues at Camarby have gathered monitoring data over many, many years to enable us to predict quite accurately when certain coral species will spawn. These predictions are essential for anybody who wants to work with coral spawn or to get prepared and to be in the water at the right time. The October spawning involves quite some species, though you never can be sure that a particular species will spawn on a particular night. Tanya and I are looking forward to cruising around with you on the reef and see what will happen. Every species has its own pattern and time to spawn. This mountainous star coral is a hermaphroditic species like most coral. It produces eggs and sperm in each single coral polyp. Prior to a spawning event, the gamete cells are put together in bundles. The species will spawn between 5 to 8 days after full moon from July to October. It releases its egg sperm bundles within a second, which is very impressive to witness. However, the bundles are visible for several minutes in the mouth of each coral polyp as the coral gets ready to release them. And this pattern allows us to know which coral colony will go next. However, the massive starlet coral, for example, is what we call gonochoric. This means that there are male and female colonies. Here you can see a male releasing sperm clouds. I witnessed once a mass spawning event of this species in front of the Curacao Sea Aquarium. It was unbelievable to see the crystal clear water turning into a murky soup within minutes and turning quickly back into crystal clear after the corals were done. And other colonies of the species are female, like this one, releasing eggs directly in the water column like a little confetti party. I'm also looking forward to this dive expeditions, since Curacao is known for its biodiversity. And by the way, all these pictures are taken in Curacao. From a scientific point of view, Curacao is a very interesting location because you find quite pristine reefs next to degraded ones. Those are usually in front of villages. Tanya and I are planning to take you to one of those places to see how our coral outplants are doing under such challenging environmental conditions. Right in front of the Curacao Sea Aquarium flourishes a healthy alcon coral population. You can see some of them here in the background. Some of you will know that the alcon coral is a keystone species for Caribbean reefs that protects coasts from heavy storms. However, the alcon coral is one of the most endangered coral species in the world, with more than 98% of the population having disappeared in the past 30 years. The alcon coral has been a focus for SECORE and our partners in the Caribbean. Already in the early days in Curacao, we produced babies at the Sea Aquarium and outplanted them on the breakwaters in front of the aquarium. The outplants grew quickly, like this juvenile coral, which is about 3 years old. I'm excited to getting back to Curacao to see how our very first outplants are doing. I hope that you will consider joining Diventures and me or us for this exciting dive expedition. And if you are not able to join us, I hope my presentation has piqued your interest in our work to restore and conserve coral reefs. And with this, I want to thank you for your attention and hand it back over to Sabrina. Thank you.
Thank you, Dirk, for that great presentation. If anyone has questions, please put them in the chat and I can read them to Dirk, or feel free to unmute and ask them. I also will put the link in the chat to the trip that we are doing together. So if you're interested, you can click on the link and find out more. So feel free to put any questions in the chat or just unmute and ask. Okay, we do have one question. Has anyone tried freezing coral gametes?
Has anyone tried breeding coral gametes?
Freezing.
Freezing. Oh, yeah. Oh, okay. That's good. That's a great question. Yes, indeed. So cryopreservation has been developed since many years, and I'm quite proud to say that one of the leading scientists in this area has been with us in the very early days. Before we went to Curacao, we were focusing on Puerto Rico, and Dr. Mary Hagedorn was there to for the very first time cryopreserve sperm cells, and ever since has made a lot of advances. So yes, in the meantime, there are even gene banks or frozen coral banks with gametes. Actually, our partners at ComRB have managed to use cryopreserved sperm from Puerto Rico and Florida to fertilize eggs in Curacao. Yeah, yeah. So it's possible and important.
That's really cool. Okay, another question. I have seen pictures of the trees used in coral fragmentation. How does that relate to SECORE's work?
Oh yeah, that's great. So very important to say is that there is no ultimate solution or technology to restore coral reefs. It's more like we're looking at developing a toolbox for restoration practitioners, for reef managers, where they can choose the right tool for a specific situation. And actually, so what you're talking about is these trees they harbor coral fragments, small pieces of coral that are taken from a donor coral, and they are genetically identical, and they're allowed to grow, and then they're outplanted on the reef, which is another way besides sexual reproduction. This is asexual. Another effort, and what we are doing is we are collaborating with, most of our partners are having such trees and work with coral fragmentation, and we are using those trees increasingly to develop broodstock. Yeah? So to let those coral, leave them longer on the trees so they can reach maturity, and then we are actually, and I haven't included all this, so there's much more going on in regards development of tools and technologies. I think those of you who joined the previous webinar at Diventures might have seen those. So we basically developed big collectors that you can pull over a tree like a condom, and then all those gametes, they drift up and are collected in a big sample tube, and that is a very efficient way to collect many gametes, eggs, and sperm cells from many colonies.
Thank you. Great information. We have another question. Dr. Alex Broski says, "What a fantastic presentation. I've long been a supporter of SECORE. And first, I want to take this opportunity to thank SECORE's Communications and PR Officer, Dr. Karen." I don't know if I said that right, but for serving as a coral restoration technical reviewer for my new book, Beneath the Blue Planet. Your point about scalability, oh gosh, my mouse, is spot on, but not well recognized within the recreational diving community. I was wondering if you ever see a time when the technology will be refined to a point where it can be implemented by those with little formal technical background, such as dive professionals and resort operators.
Yeah, absolutely. Yeah, great. Thanks a lot for the compliments, Alex. Yeah, absolutely. I mean, this generally speaking, we at SECORE, we see ourselves as the connectors between science, engineering, and implementation, and we provide all that knowledge to others so that they can take care of their local reefs, and including hotel businesses that increasingly recognize, hey, we have to take care about nature because that's why we get our guests. And the diving industry, of course. Yes, so we are, as I said, for example, we work, one of our most advanced trainee, who is actually finalizing the trainee program this year after 5 years, is as a nonprofit in the Dominican Republic called Fundemar. And the Dominican Republic has really serves really as a blueprint for blue economy and connecting economic interest with nature conservation, coral reef conservation. And around the Dominican Republic, several nurseries were created that are managed by nonprofits that are associated with certain hotel businesses. And there's a great community effort done to involve not only the local community, but also guests that go to the Dominican Republic to enjoy the underwater world and to help with preserving it.
Awesome. Okay, a couple more questions. How does the scalability of sexual reproduction of coral compare to other methods?
There's a great potential because, as I think I've mentioned in the presentation, if you want to increase scale, you have to reduce costs, right? And with the tools that were applied for many years, especially fragmentation, is really easy. You know, you break a piece off, you glue it on a substrate, then you wait a little bit till it's regrown a bit, and then you glue it on the reef. So it's very easy, but there are two disadvantages. You produce clones, so they don't add to genetic diversity, and they do not ultimately or immediately add to genetic diversity and increasing resilience. The other disadvantage is that you really deal with every single coral a couple of times manually, and that limits the scalability. However, again, we are looking at combinations, applying fragmentation and sexual reproduction in a way to make it scalable. The big advantage with sexual reproduction is that, you know, once you know what a coral likes, what the larvae like or the embryos to develop to larvae, and what the larvae like to settle on, and what conditions they need, then it doesn't matter if you provide those conditions to 100 larvae or to 1 million. Yeah, so that it's very scalable, because we can work in units where we handle a couple of hundred substrates as a unit. Yeah, and the outplanting is also very scalable, because we're seeding. We're sowing those substrates with the corals on the reef. It doesn't work for all conditions. So those reefs that are very exposed to, you know, surge and waves, we are looking now, and this is really what happens at the barrier reef along the coast of Mexico. Is that the exposure is so strong to waves that we need to find a more efficient, we need to attach the substrates, and we are looking now in how can we do that cost and labor, or reduce the labor, reduce the time that we need to fix one substrate, and that will allow in the second place to scale it. Yeah.
All right. Okay, the next question. You mentioned using sperm from Puerto Rico and Florida in Curacao. I realize there are regulatory issues, but what can be done to exchange gametes from different countries to increase genetic diversity in each country's reefs?
Yeah, so it's a very controversial topic, and I remember a conversation I had with the leading geneticist for Caribbean corals. Her name is Professor Dr. Ilyana Baums. And I think it was like, yeah, when we were working in Puerto Rico actually together almost 20 years ago, and I said, hey, why don't we get you know, larvae over from Puerto Rico to Curacao? And she, back then, basically the science community was very much against it, and now it has changed because unfortunately the conditions have worsened, and now it's really making a decision between losing that species at a place or preserving it by introducing genes or gametes from other locations. And it's, you know, it's, yes, there is a regulatory and a permitting issue here for sure. That's very important. And those corals that were produced in Curacao using the sperm from Florida and Puerto Rico, some of them went to the Florida aquarium, and they're still kept there in captivity, because there is, you know, there's now a debate going on. Wow, and they're doing really well. Yeah, should we outplant them in Florida or not? Yeah, so it's a very critical and very important discussion.
Okay, speaking of engineering, I've noted the substrates have undergone significant redesign over the years. Can you provide any insight on what has driven these changes?
Yeah, absolutely. A very good observation. So the very initial substrates were based on. I had the idea like many years ago that, you know, it would save a lot of time and increase scale if we could sew those substrates, and I thought of a tetrapod design. And back then I asked my brother, who's a professor for mechanical engineering, if he would create those, and he did, and it was a big success. And then as the team grew with SECORE, we got an engineer on board who was building partnerships with other engineering, faculty, industry. And we also have a contact, a partnership or collaboration going on at the University of Miami with the Sustain facility, which is basically the biggest storm simulator in the world, where we expose those substrates to certain wave and storm conditions and to see how they behave. And we also monitor the survival of the settlers on different designs. There's a cost aspect. So we are currently working together with a partner in Saipan who has had the idea to produce very small substrates, because they would fit very well the reef topography in the Micronesian region. So there are certain aspects like cost, attachment ability to the reef, but also increasing survival of the coral. And we are not looking at one ideal design. We are looking at providing, again, an array of designs, shapes, sizes for different reef conditions and different coral species.
It's really cool. All right. Does anyone else have any questions? Okay, let's see. There's another one. Thanks a lot for the great talk and your efforts. Maybe a naive one. Are there also restoration efforts for soft corals possible and necessary?
Yeah, interesting question. So yeah. So ideally, you would target any coral species or any species, any organism in the reef and help to preserve it and restore it eventually if it's threatened. Of course, that's not possible. That's also not very economic. So what we are doing is we are really focusing in the Caribbean on key reef builders, on those coral species that provide the framework and the topography and the complexity of a coral reef that will then allow, in the second place, to for other organisms to repopulate this restored area. And we are still a long way to go from restoring a reef. And actually, now if anybody here of you would be more into the definition of restoration and rehabilitation, restoring, and we are using the term restoration because it's so well known in the public and easily understood. But actually restoring would mean, restoration would mean putting something degraded back in its original state. And that is not possible with coral reefs. It is not possible. What we are looking is restoring the ecological and the economic value of coral reefs. That means restoring the reef framework in a way that it would help to maintain biodiversity and attract other species to repopulate the area, and then to provide all those very important economic values like, coastal protection, fisheries. Many coral reefs harbor a lot of fish. And then, of course, the value for tourism, for example.
All right. How do the corals show when they are ready to spawn?
Yeah, I was waiting for that question. So it's it is very interesting. Actually, in Curacao, when I jump in the water, and and this is in front of the sea aquarium, they have 2 entries through the breakwater, which is really convenient. And I don't see the corals right away, because I have to pass that in between zone. I hear it. I hear that this is the night. So there's a lot of noise in the water, a lot of clicking, and yeah, I don't know how to call it, because all of the creatures, especially crustaceans, so crabs and shrimps, they're getting ready for the feast, and you can hear it. So which is a very good sign. Sometimes when when the spawn is smaller, but still something is happening, you don't hear it, because they know they get ready for the next night or so. I don't know. And then when you look at a specific coral, you see. So we know now, after people like Valerie doing a lot of research in understanding the reproductive behavior of many species. I think we're studying at least 10, perhaps 15 species right now. And there are some, there's this brain coral that we call Coprophylia natans, that spawns at dawn. No, at dusk. Still dawn, yeah. So before sunset, right before sunset, if I'm correct with the terminology. And what you see is that butterfly fish, they gather and they swim around in small groups, and they check the coral. They check those brain coral, and they swim around, and then we know now it's going to happen. And that coral, it just shows the egg sperm bundles very, very slowly, very quickly, just before they are released, because the coral sort of knows that if I expose those egg sperm bundles too long in the mouth, those fish will feed on them. So they swim around, and then these egg sperm bundles are released very quickly and stop again, because then the fish leave, and when the fish left, then they start again. It's crazy. Other, like the mountainous star coral, you see the egg sperm bundles shining through the tissue for up to 20 minutes. So you really can see patches, or ideally the whole colony suddenly has these orange color, these orange small bubbles in the just at the mouth or below the mouth. And with that species, it's really tricky, because they sit there forever, and suddenly, bam, and they're all simultaneously released. And usually the condition in those spawning nights, are the water conditions are really calm. So you have this big mountainous star coral covered with those egg sperm bundles in like almost bright orange, and then within a second, and they stay there for another second, and then they slowly drift away. Fabulous. I must tell you, that was one of my core species I worked on during my PhD thesis. And I had this one situation. I was having a net, and usually you go out in a group, and then you disperse, because everybody has some nets, and then you hoover around and you look for those colonies that are staging. We call it staging. So I found two really nice ones next to each other. And I looked at one, and I looked at the other, and I thought, and I had one net only, so where do I put my net now? And I was just waiting and looking and turning, and then I turned around, and that one went off, and then I turned back, and the other one went as well, and I got nothing. And the other coral that I showed, the gonochoric one with male and female colonies, you just don't notice. You notice when it's going on, because suddenly you see these sperm clouds passing by, and it's really a chimney, and then you see like little egg cells passing by, hundreds of them, and then you know something is going on. And often how we as scientists recognize there's something going on, we see such a pattern, such a cloud, and we follow it to the origin, and then and sometimes it's a brittle star, or a tube worm, a Christmas worm, or a sea cucumber that is spawning, and that can all happen in October. So it's really, I love it just to cruise around and see what's going on, and or gorgonians start spawning, and you see the very bright eggs sitting on the polyps of those branches, and are just released. So every coral has its own pattern, and I mean, what we will do for the trip is I probably will put a few slides together, PowerPoint slides with some pictures, and then really show like, hey, this is what you have to look for. Yeah.
Yeah, I really hope nature works in our favor. So be nice to see. With only a decade until there is truly a crisis for coral reefs, how do you stay positive?
Yeah. I'm very optimistic. I think there's a lot of, certainly it's a big challenge, the climate change crisis, which not only affects coral reefs. Coral reefs are certainly the canary in the gold mine. If you look at the oceans, if coral reefs go down, it's gonna affect the whole ocean and us. So I stay positive because we make progress, and the landscape is changing. There is much more attention now and support, and there's something going on around the climate change, is addressing those climate change issues as well. It's a big challenge. Time is running, but it is just great to, in all that bad messaging, and it's all going down, we can't do anything. It's nice to show, yes, you can do something.
Yeah, we got to stay positive. All right, what is the status of sexual reproduction in the Caribbean compared to asexual restoration? What are the difficulties to implement sexual reproduction in the Caribbean?
Yeah, so fragmentation, as I said, has been carried out for a long time in the Caribbean and in other places. Sexual reproduction was studied by scientists, but I think it was actually SECORE that started to and not only us, but at the time there was, I remember one scientist who was working with us, who was looking into how can we apply sexual reproduction for restoration. And then it took quite a few years, but now every year more so we have those Facebook and WhatsApp communities where the people on the ground exchange during the spawning nights. Hey, what's going on? Are they spawning at your place? No, wow, yes, here's a big spawn. And that is increasing dramatically every year. More and more groups join, and yes, and I'm a bit proud to say that some of that, or if not most of it, is because of our training and implementation program. And nicely now we can see that the first groups that mature and leave the program still want to continue working with us, and there we are looking at working together on larger projects. For example, the partner in the Dominican Republic, we are now working on a project that is funded by the Caribbean Marine Biodiversity Fund, that provides capacity building to other countries, and in this case, it's Cuba. So the trainees are now moving into a trainer role, and that is like, of course, building a lot of synergy and helps us to get the knowledge even faster out to as many people as possible. It is more complicated, of course, than fragmentation, and that's why we believe it's really important that people follow the training. And it's also great for networking, because you get together with others, and yeah, and I think it's one of the most beautiful things of nature is to see coral spawning. Yeah.
All right. Another question. I recently attended a seminar by a researcher doing sponge restoration in the Florida Keys. Her preliminary data indicate that coral recruitment on reefs was higher on reefs with her restored sponge populations. Have any of your researchers looked at what roles sponges or other organisms might play in recruitment on your sites?
Yeah. Very interesting. I've heard about it. The background here is that the whole metabolism, the biochemistry in the coral reef is very complex, and sponges, we know by now, are very important for the recycling of nutrients, but also keeping the water clean, and to say it in a really simple way, and corals like clean water. We now at SECORE, we currently do not carry out research in that respect, but we know those researchers who work with sponges and in that area, and yeah, very, very interesting. We are looking at, for example, for effects that we can control. So it's very important aspect is restoration of other key organism groups. Yeah, so sponges might be really interesting in that regard. Sea urchins are very important. That's where we have some work going on, where we look at co-culturing or co-breeding sea urchins with juvenile corals, because the young sea urchin graze on the algae and keep the algae growth under control, because otherwise those algae overgrow those tiny coral polyps and kill them. And in the Caribbean, there is a key species that is the long spine sea urchin, Antillarium, Diadema antillarium, that was suffering from a disease a few decades ago, actually about 30 years ago, and was wiped out largely throughout the Caribbean. And that was one of the main reasons that caused the large die of the Alcon coral, for example. So we are looking now, and again, and this is great about SECORE because it's a network. So we are trying to connect those people that work on in specific areas, but we also try to collaborate with them. And we have a collaboration going on where we look at propagating this very important sea urchin, and to see how it affects the success for sexual coral reproduction and restoration.
Thank you so much, everyone, for joining today. And we also hope that you can join Dirk and I on this trip together to Curacao. Dirk, any final last thoughts?
I just want to thank you, Sabrina, Dean, all the team at Diventures. Yeah, it's great. It's just great to work with you. And I'm very much looking forward to getting to Curacao and to share the excitement around coral reefs and coral spawning in our work. Yeah. Thank you.